global to national, military to human security threats we all are facing? And what is the role of technology in this security? Then welcome to this channel, Threat, Security and Technology. In this video, we'll explore the role of synthetic biology in enhancing the security as well as creating new threats. We'll examine the complete security ecosystem from geopolitics, global to national security, military to homeland security, technologies and industry. Hello, I am Rajesh Uppal, Director, Founder of International Defense Security and Technology Incorporated, a think tank in California, US, promoting awareness on defense and security issues and technology solutions. I have also founded US-India Science and Technology for Security to facilitate US-India academia and industry collaborations on security systems and technologies. I was Director, System Analysis, Safety, Quality and Reliability in Indian Defense Research and Development Organization, New Delhi, before I moved to US. Before exploring synthetic biology, let us first understand natural biology. The most basic building block of all living things is a cell, and a human body contains trillions of cells. Cells has many parts, each with different function. In the middle of the cell, there is a nucleus which serves as cell's command center. It gives direction to the cell to grow, to mature, divide or die. Inside nucleus, there are large molecules known as deoxyribonucleic acids or DNA, which contains biological instructions which make each species unique. Most of the DNA is present in the nucleus and it's called nuclear DNA. The organism's complete set of nuclear DNA is called its genome. During reproduction, the nuclear DNA is passed from parents to the offspring. And organisms inherit half of their nuclear DNA from male parent and other half from female parent. Chemically, DNA is made up of building blocks called nucleotides. Nucleotides have three parts: a sugar group, phosphate group, and one of four nitrogen bases. The four nitrogen bases are adenine A, thymine C, guanine G, and cytosine C. It is order and sequence of these nitrogen bases which determine what are the biological instructions contained in a strand of DNA. For example, if one sequence ACTT, GTT instructs for black eyes, another sequence ACT, GCT may instruct for brown eyes. All persons have got same 99% of DNA sequences. It is only rest 1% which makes each person unique. That determines how tall he is, what is the color of his eyes, and how likely he is to have certain diseases. The physical structure of DNA is like two strands wrapped around each other in a ladder-like structure called double helix. These two strands are joined together by a pair of nitrogenous bases by a hydrogen bond. The pair has a rule that nitrogenous base A will pair with T and nitrogenous C will pair with. The unique structure of DNA molecule plays very important role during cell division. Before cell division, this double helix splits in the middle and forms two single strands. Each of these strands then serves as a template to form two identical double helix molecules, each replica of original. This is essential so that the new cell contains exactly same copy of DNA as the old cell. The same structure is also used for making proteins. In this process, the DNA molecule unwinds and a single strand is used as a template. This template is then copied to mRNA. mRNA is another molecule that carries instructions for body's protein making machinery. The process through which DNA is copied to mRNA is called transcription, and the process through which mRNA builds the proteins is called translation. Proteins are useful for many body functions such as building cells, moving muscles, and digesting food. The humans have billions of DNA molecules per cell. For efficiency, these molecules are tightly packed into chromosomes. Humans have 23 pairs of chromosomes, one set inherited from mother and another set inherited from father. Our sex chromosome, denoted as X or Y, determines whether a person is a male or female. The chromosomes are divided into short DNA segments called genes. In humans, genes may vary from few hundred to millions of DNA bases. The genes determine all our hereditary traits. They are also responsible for producing one specific molecule, which is usually a protein. The human genome contains about 3 billion bases, 20,000 genes on 23 pairs of chromosomes. This complete collection is also called its genotype, contain all the instructions for building and maintaining human being. Whereas the set of observable characteristics that result from this genotype is known as its phenotype. Let us now explore synthetic biology. It is an engineering approach to the biology. Its aim is redesign of natural biological systems for useful purposes, as well as design and construction of new biological parts, devices, and systems. How it does? Any organisms sensing, metabolic, and decision-making capabilities are encoded as unique DNA sequence within their genome. These DNA sequences determine what a cell grows, what goes inside it, and what it produces. By changing these DNA sequences, we can change these cellular functions and thereby engineer them. Let's now consider some of the tools and technologies of synthetic biology which allows us to engineer biological systems. The first technology is to read DNA or DNA sequencing, which allows us to determine the order or sequence of DNA base pairs or biological instructions contained within a strand of DNA. A difference from expected gene sequence is known as variant or mutation. By comparing the expected normal and mutated DNA sequences, scientists can diagnose various diseases, including cancers, and deliver personalized human care. The rapid speed of DNA sequencing technologies has enabled sequences of complete genomes of different species and types of life, including microbes, plants, animals, and the human genome. This technology is also helping the growth of genome repositories. One such example is International Nucleotide Sequence Database Collaboration, or INSDC database, which has now grown to more than 100 billion base pairs. These databases contain information on natural organisms from which synthetic biologists can generate parts and devices. The synthetic biology products have also multiplied since DNA sequence technology has become better and cheaper. The second is the gene editing technology, And CRISPR has become one of the most popular technology because it is fast, cheap and easy to use. CRISPR can locate, cut and replace DNA at a specific location, thereby modifying the function of the gene. It uses modified RNA sequence that recognize the DNA sequence in the genome and then binds to it. RNA also attaches to Cas9 enzyme which can cut the DNA at the target location. The CRISPR is also enabling gene therapy which adds, deletes or corrects genetic material to treat the diseases. The next technology is DNA synthesis, which is the natural or artificial creation of DNA molecules. We have already seen natural creation. During cell division, the DNA molecules helix splits into two parts and then each part serves as a template for duplicating the base pairs. This natural synthesis process is also known as DNA replication, as it replicates and makes copies of itself. Another is the polymerase chain reaction or PCR, which amplifies the DNA sequence. A short DNA sequence called the primer is used to select the portion of genome which is to be amplified. The temperature of the sample is then rapidly lowered and raised so that the DNA replicating enzyme can copy the target DNA sequence. This process can create billions of copies of target sequence in just few hours. This PCR technique is also useful for genetic fingerprinting and criminal forensics. Traditional DNA synthesis technique were chemical that required toxic chemicals and generated hazardous waste. Further, they could only synthesize short DNA or RNA molecules called oligonucleotides about 200 bases long. To assemble even a small gene, scientists used to synthesize short segments and then stitch them together. Longer sequences resulted in errors and low yield. Therefore, traditional gene synthesis technique was slower and expensive, especially for long strands. The new DNA synthesis technique is called enzymatic DNA synthesis or EDS technique. This technique is based on DNA synthesizing enzyme found in the cells of the immune system. This enzyme is able to add nucleotides to an existing DNA molecule in water where DNA is most stable. The improved precision of this technique allows to synthesize strands of several thousand bases long, about the size of a medium-sized gene. This technology has enabled development of DNA printers. Earlier scientists used to search for DNA in nature, cut the DNA from existing organisms, and then insert it into host organisms using a cut and paste process. These new DNA printers can just synthesize DNA from scratch using any code you like. You don't need to find now DNA in nature. You can order it online. There are also many commercial companies which are providing DNA synthesis service. Most synthetic biology companies are producing artificially DNA codes that can be inserted into microbes, plants and animals, forcing them to produce industrially useful compounds. DNA replication property can then allow this to scale up to millions of programmed cell factories, filling up big industrial vats. Synthetic biology, therefore, has turned bioscience into future manufacturing paradigm through which companies can engineer and produce infinite quantity of things cell by cell from scratch. These bioengineered microbes, plants and animals can then develop new pharmaceuticals, break down pollutants, kill cancer cells, detect toxic chemicals, and even generate hydrogen for post-petroleum economy. Let us now consider synthetic biology geopolitics. Biotechnologies, including synthetic biology, are going to be foundational in 21st century. Synthetic biology is already a multi-billion dollar industry with wide range of application: drugs, health, energy, chemical, food, agriculture, and other sectors. By end of this decade, it is predicted that synthetic biology products will account for more than 1/3 of the global output in manufacturing industries and more than 30 trillion in value. It is also predicted to transform defense and security by producing on-demand production of novel biofuels, cells, food, sensors, coatings, whatever the military needs. The economic and military advantages are driving intense synthetic biology race among countries led by US, Europe led by Germany, Japan, Canada and China. Currently, US account for nearly 40% value in global output, which is 3.3 billion estimated in value. However, China is pursuing an aggressive strategy to become world leader in biotechnology, driven by its aging population, health and military needs. It is investing heavily in biotechnology. It also has dedicated strategies for providing thrust in commercial and military applications. For example, biotechnology is a key strategic priority in China Made in 2025 initiative and also current 5-year plan. Let us now explore the role of synthetic biology in global security. Synthetic biology can address many global challenges such as climate change, energy security, food security, health care, and more. It can address global warming by developing products that are more sustainable, use no fossil fuels, less land and water. It can create artificial leaves which can synthetically carry out photosynthesis process. It can fight pollution and other environmental hazards. It can detect toxins such as arsenic, cadmium, mercury, nitrogen, phosphorus and other heavy metals and respond appropriately. It can healthcare sector. Synthetic biology is leading to speedy development of new vaccines. For example, during COVID-19, mRNA vaccines were developed within a year. Other areas may be low-cost diagnostic tools, biosensors that can reside into your body that can detect and fight diseases like cancers. In agriculture, nitrogen producing microbes can replace chemical fertilizers. Engineered microbes can be used to produce food that is more sustainable, healthier and ethical. Then in energy sector, they can be used to produce biofuels more efficiently. For example, biofuels based on microalgae have got high fat and carbohydrate content, grow rapidly and are also robust. Space synthetic biology is indispensable for future Moon and Mars mission and permanent settlements there. Human mission require supply of food, shelter, medicine and materials. However, cost of carrying them is unsustainable. Therefore, researchers are developing synthetic biology methods to produce them in space. For example, astronauts may carry cells of fungi and bacteria which can be programmed with synthetic DNA to produce materials such as bioplastics. These materials can be then fed to 3D printers to produce anything what astronauts may need, from medicine, food, medical devices, and etc. They can also take DNA printers with them. Scientists from Earth can send instruction to these DNA printers to create new microbes that can then produce novel chemicals and materials. However, there are many challenges because environmental conditions on Mars surface are very harsh, such as low temperature and pressure, high ultraviolet radiation, then little moisture, oxygen prevent the microorganisms to grow there efficiently. Synthetic biology can then even alter the microorganisms genetically and make them more space worthy, more resistant to space radiation and heat. Now let's consider role of synthetic biology as a global threat. Synthetic biology is a dual use technology. It can be used both to benefit and harm people, animals, environment, and even nations. First, is a threat to agriculture and farmers. It is rapidly replacing naturally grown compounds with lab grown food and consumer ingredients such as synthetic saffron and vanilla. This is threat to farmers' livelihood and entire ecosystem of forest and agriculture fields. It is also displacing agriculture. Growing vats of engineered yeast or algae requires vast amount of corn and sugarcane plantations. Synthetic biology can lead to three major global threats: pandemics, biological warfare, and bioterrorism. Biological warfare is the use of biological weapons by nation states in a warfare which is prohibited by Biological and Toxin Weapon Convention. Bioterrorism aims to create disruption and panic among the population. Synthetic biology can enhance the threat of pandemics and epidemics. Earlier, we had SARS, MERS, H1N1, and Zika. Now we are experiencing COVID-19 pandemic, which is leading to grim milestone of 6 million deaths. According to consensus, COVID-19 was not a bioweapon and probably not genetically engineered. The more probable cause was it was laboratory associated bioaccident caused by laboratory poor biosafety standards. However, it has demonstrated that even a single viral strain caused by bioaccident can create devastation and havoc for years. Synthetic biology can expand bioaccident scenarios. What if an algae bioengineered to create gasoline escapes in rivers and oceans and produces self-replicating or self-generating oil slick? So now we consider bioterrorism, which is the deliberate release of virus, bacteria or other agents in order to cause illness or death in people, animals or plants. Synthetic biology, by modifying or editing the genome of organisms, enables scientists to create new bioweapons. We saw earlier that DNA printers allow us to build DNA from scratch with any DNA code you want. You don't need to find DNA in nature anymore. Scientists could employ DNA or RNA of pathogenic viruses and bacteria, which are freely available online, to create new bioweapons. In fact, assembling of entire genomic infectious genome has already been demonstrated in the case of poliovirus and Spanish influenza virus. Synthetic biology can also enable development of superpathogen, which could eradicate entire civilization. It could also enable development of new kind of bioweapon which could target only selectively certain population or races. Thus far, the bioweapon production has only been carried by nation states. However, as the knowledge, skills and tools of synthetic biology are getting increasingly available, it is expanding the range of threat actors and also decreasing the time required. Still, production of bioweapons faces many challenges. First, it required knowledge, specialized expertise, teams, infrastructures, and etc. Also, it is difficult to maximize all the desired characteristics simultaneously, such as virulence, transmissibility, or stability. There are also process challenges such as scaling up, storage, and dissemination. The scaling up requires a gradual approach, starting from laboratory samples to pilot stage production to final full stage production, which may take many years. The dissemination of biological agents also faces many technical challenges. Adding biological agents to urban reservoirs faces challenges of dilution, chlorination, and filtration, which can kill most natural pathogens. Similarly, adding bacterial contamination to food gets killed by practices such as cooking, boiling, and pasteurization. Unlike chemical agents such as VX nerve gas, which can be absorbed through skin, biological agents need to be inhaled or ingested to cause infection. Therefore, most effective way to disseminate biological agent is through respirable aerosol. That is, large invisible cloud of micron-sized infectious droplets which remain suspended in air for a long time and can be inhaled through large number of people. Again, the challenge is to develop such a sophisticated apparatus which can generate such particle size agents and also in large enough concentration to affect a large area. Moreover, most of these biological agents cannot survive more than 30 minutes in a broad sunlight because of their sensitivity to ultraviolet radiation. Therefore, it is limited to nighttime attacks. Therefore, the possibility of terrorists carrying large-scale bioterrorism attack is of remote possibility at present. Synthetic biology will also play a very important role in national security. It will enable development of new materials, green fuels, medicines, including vaccines, then engineered insects for pest control, to better crops which can grow in drought. It can also enhance national threat in many ways. The knowledge and skills to develop and disseminate biological weapons is becoming increasingly accessible. The reagents and tools of synthetic biology are becoming available as commercial kits, which becomes easy for biohackers to acquire. The well-characterized biological parts are becoming increasingly available from open source genome registries. Then the assembling of small fragments into larger ones to complete genome is also becoming simpler and available across the globe. There is also threat of hacker culture, increasing the risk of reckless or malevolent experimentation. We saw earlier that the risk of terrorists carrying a large-scale bioterrorism attack is remote. However, of them carrying a small-scale bioterrorism attack is still possible in the near term. For example, they could carry a biological weapon attack in an enclosed space such as building, shopping mall, subway station, or in sports arena. As we saw, Such an attack will be in the form of respiratory aerosol, and it may affect thousands of people. Another threat would be recreation of known pathogens such as Ebola virus in the laboratory to carry out a bioterrorism attack. The other is the creation of novel threat agents which could target groups or important personalities based on their biological features. There is also threat to agriculture and food. The agriculture bioterrorism targets food and livestock during their production, harvesting, storage, and transport. It is a part of economic warfare. The economic warfare targets the country's agriculture and ecological infrastructure by use of a biological weapon. Biodefense strategy therefore needs to be comprehensive to cater for all the threats from biowarfare to bioterrorism, agri-terrorism to bioaccidents. Some of its elements are developing biodefense capabilities to prevent occurrence of bioaccidents, preparedness to limit their impact. You need national network of sensors to rapidly detect and characterize these bioaccidents to people, animals, food, and environment, then rapidly respond to limit their impact, and finally recovery to restore the community environment. During COVID-19, we saw shortages of medicines, including vaccines and equipment such as protective masks and ventilators. There is need to establish manufacturing capability to rapidly manufacture such items during emergency. There is also need for coordination between government, academia, and industry. Let us now explore the role of synthetic biology in modern battlefield. Biology is considered the future of warfare. Some also consider it to be new domain of warfare. We saw in COVID-19 that pandemic affects military. It degraded military capability. The crew of third of the US military aircraft carriers got affected. It degraded military preparedness through limitation of movement of personnel and cancellation of many training exercises. Synthetic biology can enhance military capability by enabling both offensive and defensive applications. Militaries are planning to use it for performance enhancement, development of new sensors and materials. One program aims to engineer marine microbiome to detect the chemical signature of diesel electric submarines and release biomarkers into the environment. This will be then detected by nearby drones and communicated back to the forces, which can then send anti-submarine units. The use of biosynthetic fuels in jet and diesel engine can increase their range and fuel economy of aircraft, ground vehicles and ships while reducing greenhouse emissions. It can also be used to develop adaptive camouflage, cloaking devices, then stronger, lighter and self-healing body and vehicle armor. For example, scientists have created spider silk, which is lighter, stronger, flexible, and can be used in body armor and aviation composites. CRISPR and other biotechnologies can also enhance or degrade the performance of soldiers, making them smarter, lethal, and enhance their endurance. The protein of microscopic animals, nicknamed water bears, can protect human DNA from X-ray radiation. Human cells cultivated with newly discovered protein dubbed DSUB or damage suppressor found to only decay only half as much as normal cells when exposed to radiation. In future, it can also protect human from space radiation. Our body's microbiome consists of collection of the genes of all the microbiotas. And what are microbiotas? They are the microorganisms that are present on our body and inside our body, including viruses and bacteria. For example, synthetic biology can edit the gut microbiome and then which can improve the digestion and cognition. These are also very important for warfighter performance. It can also harm the humans. We have heard of cyborgs, which are hybrid of machines and organisms. In the future, synthetic biology will enable new generation of cyborgs that will be biocyborgs. The offensive purpose of synthetic biology will be employing biological weapons which are hard to detect, trace or control, and against which there is no treatment. In the battlefield, biological weapons will be very difficult to control and predict. And it is likely that troops on both sides will get affected. We saw earlier that at present there are many challenges for terrorists to carry out large-scale bioterrorism attacks. But what about state-backed terrorists? Can they carry out large-scale bioterrorism attacks? The Biological and Weapon Toxin Convention, BWTC, bans the biological and toxin weapons by prohibiting their development, production, acquisition, stockpiling, and transfer and use. However, the effectiveness of this convention has been limited because of lack of formal verification body monitoring its compliance. Some countries may continue to engage in biological weapon dual use research activities and thus fail to comply with BWC. They can access the caches of incompletely buried or destroyed biological weapon materials and transfer them to other nation states or terrorists. The advances in CRISPR, Cas9, genome editing, nanotechnology, and biomimetics also enable weaponization of non-biological weapon materials, which falls outside BWC. China was first to use CRISPR on humans and created first gene-edited babies. China's civil-military fusion strategy also allows them to readily access the developments in civilian biotechnologies. DARPA has launched multiple programs to protect soldiers from accidental or harmful effects of genome editing technologies. These include protecting the soldier from getting affected in the battlefield, rapidly detecting the pathogens, developing vaccines, and quickly ramping up vaccine production. For all of us, most important is human security. That is freedom from threats to our lives, safety, and rights. On one hand, synthetic biology can generate more effective therapies, personalized medicine, such as targeted chemotherapy. Synthetic biology products are more durable, generate less waste, and healthier for human generally. However, progress in synthetic biology also raises many questions in bioethics. For example, DNA sequencing enables individuals to know about their genetic traits such as ancestry and carrier status of various genetic diseases. What if insurance agencies raise their rates after knowing the predisposition of certain individual to diseases? What if employers treat individuals differently? At present, Genetic Information Non-Discrimination Act (GINA) of 2008 prohibits insurance agencies and employers to discriminate based on genetic information. However, experts point out that GINA does not cover life insurance, disability, and long-term insurance policy. Then there are questions of privacy and security. For what purposes genomic data should be used? And who should oversee that this data is being used properly? What if hackers steal that data and use it to blackmail the individuals? Similarly, using CRISPR for extreme genetic engineering of human performance enhancement of children and soldiers also has many effects. These risks include inaccurate or incomplete editing of cells of human embryos. How a synthetic organism will grow, adapt, change, or behave is anybody's guess. It is very difficult to predict what these genetic alterations will have on populations. Once introduced into the population, it is very difficult to remove them. And also, it won't be confined to any single community or country. Therefore, most experts agree that there should be process of continuous monitoring and assessing the developments of synthetic biology. The regulators also need to match the speed of synthetic biology industry. Many individuals and organizations have already taken up challenge of biosecurity. Thanks for listening. In future videos, we will continue to explore various threats, security, and technologies. You are welcome to suggest topics of your interest in the comments below. I would be happy to address them first. If you want to participate in this fascinating journey of threat, security, and technology, please subscribe.